Hello, I'm İlkay Altıntaş. I'm the chair today for the invited talks session this time. Our first talk today is Stephen Smith. Stephen is an associate professor in the Department of Ecology and Evolutionary Biology at the University of Michigan. The Smith Lab Group is primarily interested in examining evolutionary processes using new data sources and analysis techniques. Today, he's going to talk to us about reconstructing evolution with large data set and the computational challenges related to that. Stephen?
Thank you. Thanks. Okay, so I have been to several of the invited talks, and I think I'm a bit of an outlier here because I'm a biologist who develops HPC tools to answer the questions that we have to answer. But as a result, I'm going to give you some biological knowledge that hopefully will get you through the talk. So, did you know that there are plants that live in Antarctica? There are actually two species of plants that live in Antarctica. It seems strange considering how much ice there is. They happen to be closely related to cacti, which seems strange considering they're living in really dry, hot environments. Those are both closely related to carnivorous plants. So we've got Antarctic plants, cacti, closely related to carnivorous plants. All of these are closely related to beets. So that sort of gives some idea of the kind of complexity that we're dealing with. And that purple pigment that you associate with beets, we associate with some colors, the flowers and things like that have those colors, but actually the pigment might help keep insects away and is the result of an ancient genome duplication. So the way that we arrived at all of these answers is by using HPC and a lot of genomic data. And I'm going to give you an idea of kind of some of the challenges. My goal here is to basically excite you about the kind of analyses that we do in evolutionary biology and to give you some of the ideas of the challenges that we've tried to overcome. I'm going to start with the conclusions, just to make sure that the point comes across. So what basically I'm going to conclude is that larger data sets yield insights that are impossible with smaller data sets. That shouldn't be too groundbreaking. However, with more data comes more heterogeneity and complexity. So we can answer simple questions with enormous analyses, but more nuanced questions require digging in the data. So computationally, we have to find ways to conduct smaller, targeted analyses when we can. This allows for flexibility in the computation and dramatically reduces requirements, but also it reflects the biological heterogeneity. So it's computational, it's computationally beneficial, but it also is beneficial for answering our questions. Okay, so essentially this is the story of how, as a field, we've moved from doing these large monolithic analyses to these really small analyses. And the big finding is basically that this makes both computational and biological sense. All right. So I'm going to talk a lot about something called phylogenetics. So I need to give you a little bit of an introduction. Hopefully it's not too painful. So phylogenetics is the way that we understand how all things are related. So we can start with humans here. It's pretty non-controversial to say that we're most closely related to chimpanzees. Now, we would draw this kind of diagram here. There's a lot of information hidden in this. So at that internal node, we assume that there was a speciation event. So there was a lineage that split into two lineages millions of years ago. So there's some putative ancestor. Underlying that, there are populations, there are individuals, there are genomes. And so we also assume that along these lines, we have extinct lineages that we don't see anymore. So we're thinking Neanderthals, Australopithecus, that kind of stuff. Now, this isn't exactly accurate. Chimpanzees are more closely related to bonobos than they are to humans. So this gives you an idea of sort of, phylogenetics and what we do. Now, of course, this is a part of the mammalian phylogeny. We are all mammals. So this fits into some larger phylogeny there. All right, so what we're looking at here are phylogenetic trees. They're visual representations of the relationships between species, taxa, individuals, or populations. You would know them as directed acyclic graphs. So if you came to a talk wanting to look at a lot of directed acyclic graphs, you came to the right talk. So these are as old as Darwin. The only figure in the origin of species is a directed acyclic graph. It is a phylogeny. Darwin talked about these actually 20 years prior in his notebook. One of the few figures that he draws in his notebook is also one of these. So these are essential to the biological sciences. So this is trying to get at the tree of life that represents how all species are related, and we're talking about 2.5 million species, and we don't, as a biological sciences, have one of these yet. We don't have a complete tree. So a fundamental goal of biological sciences is to build that tree. Now, somebody like myself is interested in building it because we'd like to know how things like this widow bird, or this crazy beetle that looks scary and gross, or this velvet worm, or this chimp are all related. However, they also have essential practical uses for human evolution and for other aspects of, say, epidemiology and stuff like that. For example, we use phylogenies to better understand cancer. The Ebola outbreaks generally are always published with a phylogeny of the outbreak. HIV is studied this way. Importantly, for a conference like this, I assume beer strains are studied by this method. And we can also look back in time at things like dinosaur genome size to actually better understand these with phylogenetics. Also, whether it's advisable or not, people do get them as tattoos. We change these results pretty frequently, so it's best to have a very good relationship with your tattoo artist if you're going to do this. Nevertheless, how do we actually build these? Well, hundreds of years ago, there was taxonomy. You had people like Linnaeus. They would go out, collect different specimens, and they would try and associate them, do their own kind of machine learning clustering algorithm, and put them into different groups. Later on, we sort of formalized a technique to do this, and you basically might take a group of organisms, say this group of 9 organisms. You might identify some characteristics on those and code those characteristics as presence, absence, or some sort of thing like that, and then basically search for the tree that best describes that data. So search for the directed acyclic graph that best describes that data. So what do I mean by that? Well, you might take an individual character, look at it on the tree, count how many changes there are, and say, oh, okay, this tree best describes this data. Now, this is something that an audience like this should well understand. This is an NP-hard problem. The number of possible trees is enormous, and so searching around for these trees is extremely difficult. Nevertheless, soon after this, say in the 90s, we started to sequence DNA. So we look at this data much in the same way we used to look at the morphological data. We can still line it up like we can the morphological data, and we can still search for the best tree. But cracks start to arise. So if we looked at morphology, we end up suggesting that elephants are more closely related to manatees than they are to sloths. If you sequence the DNA, you actually say elephants are more closely related to sloths than to manatees. Okay, so that's something we're going to come back to in a little bit. Now, the study of phylogenetics can be summarized in a few slides here. If we imagine that this is the tree of life, it really looks something more like this. But bear with me, if it looked something like this, basically the history of phylogenetics is people either doing small analyses on little groups. So for example, this is an analysis that I did of a group of honeysuckles. And it's a group that arose in the last 20 million years, and we study it in great detail. And then there are people that still look at maybe roughly 20 to 30 or maybe 100 species, but looking really deep time. So looking, for example, at something that might be 540 million years ago. Okay, so I'm going to argue that we're going to learn a lot more about all of this stuff if we look at the entire tree of life. So my talk basically falls into 2 sections here. One, I want to talk about the computational difficulties and solutions that we have for addressing the construction of million-tip trees. And then I want to talk about how we can go into deep time and look at things that are 500 million years old. So why would we want to look at a whole tree of life? Well, let me just give you a couple motivating examples. So about 100 years ago, there was a biologist in Connecticut that suggested that maybe herbaceous plants evolved faster than woody plants. He just made this suggestion. He said that, if you had a short lifespan, maybe over the course of 10 years, you have 100 generations, whereas something that has a longer lifespan only has 1 or 2 generations. All right, so people started to look at this in the 90s with very small trees. Here you have a phylogeny where the branch length is the amount of evolution. So they have the same amount of time, but the branches that are longer are evolving faster than the branches that are shorter. And so the ones that were shorter had a slightly longer lifespan. It wasn't exactly clear. I made a set of large phylogenies that had 5,000 tips of species, and the brown branches are long lifespan and the green branches are short lifespan, and it's pretty obvious that the green branches are much longer than the brown branches. And so we can conclude from this basically definitively that herbaceous plants evolve faster than woody plants, and that's it. So now that's sort of something that we understand now about the history of life. Herbaceous plants evolve faster than woody plants. We're in the fall-winter season. There are a lot of adaptations that plants have to living in this area. For example, they can become deciduous, lose their leaves. They can also become herbaceous. A major question that we've had is whether or not they moved from the tropics and were already herbaceous or were already deciduous, and then they moved into the area, or did they move into the area and then evolve that? So with a large phylogeny of about 30,000 tips, we're able to answer that basically they were herbaceous before they moved, and then they evolved deciduousness after they moved. So that's another thing that we can say. If we look at the flora of China, we can use these same techniques, in this case with a 15,000 tip tree, and we can say that the plants of eastern China are older than the plants of western China. So we can get this kind of basic information, and primarily all we need to do is we need to increase the amount of data that we have available. And luckily, there has been an enormous effort to try and do this. So molecular data availability has skyrocketed because of sequencing techniques. Nevertheless, the amount of data that's available only represents about a third of all the species that we know. So if we're trying to make one of these enormous 50,000 tip trees or something like this, we get a lot of missing data. So you can make a plot like this, looks a little bit like the matrix, where black is missing data, green is data, and then every row is a species, and every column is a gene. The actual data set looks a little bit more matrixy, a little bit larger. Nevertheless, we can still construct a phylogeny of this, and until about 2015, this was the largest phylogeny published, 55,000 species, spanning about 350 million years, 10,000 columns of data, tons of uncertainty because of the missing data, and computational complexity. We basically can't explore the space adequately because of this NP-hard problem. Now, to construct this, it took months of CPU time, and we can calculate the amount of memory it required, basically about 67 gigabytes of memory. That's just, for the basic program that we use, we can calculate the amount of memory. So we have a few fundamental problems here. One, we need to construct a data set that allows us to get fast evolutionary rates so we can reconstruct the tips there. But then we also need things that are of low rate so we can reconstruct the base of the tree. So we kind of need to span this whole data, or this whole tree with both fast and slow rates. So we can look at how much data we actually have to inform a particular edge. Here, I've taken a large phylogeny and then just asked how much data we actually had to reconstruct each of those edges. And so red means not that much, blue means a lot, red is bad, blue is good. So most of these edges have very little information to resolve that edge. Now we can ask how much data do you actually need to resolve the edge? To give you an idea of what I mean by that, we can take a two-state 0, 1 kind of situation, and we can say that you can have one datum, one set of data where you had A and B had 1 and C and D had 0, and we can distinguish between these three trees. Does that make sense? If we had 4-state data, as we might for DNA, we can actually identify 4 different relationships. So we take the same logic and we apply it to these large phylogenies. We can say in order to resolve the edges that we had for that 50,000 tip tree, I said there was a lot of uncertainty. In order for there to be no uncertainty in those relationships, then instead of 60 gigabytes of RAM, we would need 338 gigabytes of RAM. That's the minimum amount of data that we would need. You can see in this plot basically that I have the number of states from 2, 4, and 10, and then the number of species, and you can see the increase in the amount of data that we would need on the y-axis there. So the big breakthrough, which doesn't seem like much of a breakthrough, is that we can use divide and conquer to do this in a significantly more feasible way. So if we had 40,000 species, then the minimum amount of data that we would be required to have is about 170 gigabytes of data, and the runtime of this, it would be in months. Now, if we could just make one assumption, if we could say, okay, well, maybe we understood and knew the relationship of some deep branch that would split that tree into 2 trees of 20,000 species, So the minimum amount of data in that case is 36 gigabytes, and the runtime is weeks. Well, let's just say that we had a few more edges that we could say, all right, we can resolve this part of the tree, and so we can have 4 subtrees that each have 10,000 species. Then we only require 7 gigabytes of data, or 7 gigabytes of RAM, and the runtime is in days. And you can see where I'm going with this. So we developed this method to basically apply this divide and conquer to these large trees in an automated fashion. And so we can try and say, okay, on the left side here, we have a set of relationships that we understand to be true, and we're just going to make subclades of the rest of those. And we don't need to get into how exactly we know that those are true, because we're going to talk about that in just a second. But if we do this, then we can do this divide and conquer method, and we can actually successfully make a tree. So we updated that tree that we had of 55,000 to something that had 79,000. This spans the same amount of time, but we could actually get a little bit finer scale because we had this divide and conquer method. We had more than 100,000 sites, which is more than we need, and the runtime was in days versus months. And actually, each individual analysis can be run on a laptop. So this sort of dramatically changes the thing. Not only do we get a more accurate tree, but we were able to complete it in very quick time, which means we can update it and all this kind of stuff. Now again, this is a fundamental question that we have for biological sciences. So I mentioned that this only represents about a third of the taxa that we know to be described for plants. We can expand this out to the tree of life, where I said we had 2.5 million. And we can say, well, there were all those taxonomists that have been working on this for a few hundred years. Is there a way to combine their graph so we can represent their taxonomy as a graph, and we can represent our data as a graph? Is there a way for us to combine that so that we can take all of these divide and conquer analyses and take advantage, and the species that we don't have yet from sequence data, could we combine those? And so we can. So this is basically the goal of the Open Tree of Life, which is an open source Tree of Life project that intends to basically present what is our current state of knowledge for the Tree of Life. And so through that project, we constructed this tree. So this tree actually does have 2.3 million species in it. So it's the largest tree that you'll see, because it's the largest tree that's ever been built. And this one has time in it. So if you really squint there, the time span goes to about 3.5 billion years. So we can zoom in here. So I kept showing trees, phylogenies of plant species that go around 350 million. We can zoom in there, and you can't really see it, but in that very first big bar are all the mass extinctions that you've ever heard of. So just think about how many more there were going back further than that. Nevertheless, part of this supposes that we are able to actually confidently resolve those early branches. So I've left that to the side. So we need to be able to do that. Basically, I'm talking about these ones that I assumed existed and that facilitate us to do this divide and conquer in trying to construct these phylogenies with large number of tips. So here comes the second part of the talk. So basically, the first goal was to try and expand how many tips we could get into a tree. And that presupposes we can understand how deep evolution works. And the second part is going to try and answer that second question. So, what we're talking about here are the edges that would fall in the blue area there. So these really deep edges that go back hundreds of millions of years. Now the problem is basically, so that you remember your Markovian processes here, is that we assume that DNA evolves as a Markov process. So this is relative to time. So over a short period of time, we expect few changes. Over a long period of time, we expect many changes. And at some point, that's going to saturate. We're going to have so many changes that we're not going to be able to estimate what the original base was or what the changes were. So in order to overcome this, what we always thought we needed to do was we thought we just needed to increase the amount of data that we had. So this is just one of these big data problems. We just figured in order to get all of in order to assume that we could capture those sites that had slow evolving sites, or slow evolving data sets, we had to have more and more genes. And so we increased basically the number of genes that we would try to analyze. And In part, that comes from a graph like this, where we increase the amount of data that we had. But this actually is not whole genome data. So if we take into account that blue line, this red line there is the whole genome data. That blue line is still the same data, right? So that's kind of the old way of doing sequencing. When next-generation sequencing came into play, which is developed as a result of the Human Genome Project, we get this red line. So that's how much more data we have. Now, you may or may not be able to see, but there's a little yellow line that's above the red line in the recent thing. So if you remember that, I'll tell you about that in just a second. Now, this amount of data is actually increasing significantly as we speak. So, for example, at the University of Michigan, we sequence in our sequencers about that amount of the red line sequencing every day. So that's how much data we're creating. I'm a part of some projects that are intended to sequence basically all of the organisms on Earth. So, for example, there's a project that was announced last year, this 10KP, that's intended to sequence 10,000 genomes of plants. So this is creating petabytes of data, all right? Now, we had assumed until this point that we were going to be able to do one large analysis. Now, right now, our data set sizes are in the hundreds of megabytes to gigabytes. The memory implications are something about 20 to 30 gigabytes. This isn't the one that has all those taxa, right? This is just few taxa doing deep nodes, but we're still into gigabytes of RAM and hundreds of megabytes of data. And that's without petabytes, right? So with petabytes, I mean, we're into incalculable amounts of RAM. So basically, we can't scale. So we need to figure out how can we scale this problem. There are other issues with doing these kind of monolithic analyses that I want to get into. And first, I want to remind you a bit about this conflict between doing analyses based on morphology and molecular data. Because there actually happen to be a lot of conflicts within the molecular data, and that's what we're going to talk about now. And that finding is basically going to facilitate us to do divide and conquer on those genes as well, which will solve our petabyte problem. Okay, so let me give you an example. So this is a diagram of how animals are related. So as we understand it, we have sponges diverged first from early Metazoa about 500 million years ago. They don't have any mesoderm cells. They don't have any complex tissues. Later on, we have a split that has these ctenophores. Those are the very pretty comb jelly that you see there in the GIF. Then we have later on a split that has jellyfish and bilateria. We are bilateria, but so are lobsters. Okay, so we published a phylogeny in 2008 that suggested, based on a bunch of genomic data, that this was not correct. That in fact, that sponges and ctenophores switches, right? So we moved from this traditional view, where we had the evolution of complex tissues, neurocells, mesoderm cells, all happened at one period in time, to this new hypothesis where actually ctenophores may have evolved neural cells twice, or Metazoa may have evolved them twice, ctenophores by themselves and then bilateria and jellies. So this has major implications. Now, if you were wondering, coming to this talk, what could cause a fist fight between a bunch of biologists, this is the question. So every possible resolution of this set of taxa has been proposed. So I just showed you this one in the middle. We don't need to go over the specific ones, but suffice to say, there is something that is causing a lot of conflict in these. All right, so in order to try and solve this, we assumed that we just needed more data. So what we did was we sequenced more ctenophore genomes. So in fact, we sequenced an entire ctenophore genome. That was a very large project, and tried to say, if we just add more data, are we going to be able to converge on the right answer? In this case, we also got ctenophores as sisters, so we got the new hypothesis that causes fights among people. But we also had a finding about why this might be the case. So we were able to look at the specific genes, since we sequenced the whole genome, and say that actually sponges lost the neural cells. So the neural cells still evolved only one time, and sponges just lost them. And in fact, it was those mesoderms, so the secondary cells, the things that make up other kinds of tissues. These actually evolved twice, and ctenophores, those pretty organisms, actually make them in a slightly different way than we make them. So that seems nice. We collected more data, we did one monolithic analysis, and then we're done. Well, not so fast. So nevertheless, there's still no consensus. There's still a lot of heterogeneity, and people still fight over this and find different resolutions of the data despite putting a ton of data at this, throwing a ton of data at this. So the real question is, why is this happening? Why are we getting these different answers, even though we keep gathering more data, so we would think it would converge on the right answer, but it doesn't seem to be converging on the right answer. So I'm going to go over a more specific example to give you an idea of what's going on and to give you the solution to this problem. All right, so back to those groups that I mentioned at the very beginning of the talk. This is a group called the Caryophyllales. You don't really need to remember them, but they include beets, carnivorous plants, cacti, and other really, really interesting things. So we've got carnivores here. This includes the Venus flytrap. It includes pitcher plants. It includes sundews. That one on the bottom right is a latrine plant. That little guy actually uses it as a toilet, and that's how the plant gets nitrogen. These things also live in extremely cold environments. So this would be one of two plant species that live in Antarctica. They have crazy photosynthetic modifications. So you've probably never heard of this, but they do something that allows them to survive in really dry, hot environments. So this is something that's looked at really extensively by agricultural companies to see if they can take those genes and put them into other agricultural plants. And it's called C4. Nevertheless, that's what allows cacti to live where cacti live. They also just look crazy. So there are some plants called rock plants, lithops, that live in South Africa, and they're just those two little leaves that you see down at the bottom. They also have these things that are at the top there that's called Didieriaceae. They look like Dr. Seuss plants to me. Nevertheless, they have some really cool stuff going on. So way back in the day, somebody collected some molecular data on this stuff when it was really hard, and basically they found that they could not resolve anything with this phylogeny. They couldn't resolve the tree. So that's just reflected in the really small numbers that are at the base of that thing there. So some researchers and I got a bunch of money to sequence a bunch of genomic data from this group of plants, just all over the place, to try and do a better job of this. So that little yellow line that's at the top there, that's actually just how much data we generated as a part of our project. So basically more data than is available in the public databases. So we can do that one monolithic analysis, and we can get a phylogeny. We don't need to go into any details here. It just has tens of thousands of gene regions, more than 100 species, takes a long time to run, has a really big data set. And underlying this, we assume that there's a lot of conflict, as we found with everything else. So let's just zoom into one little group, a pretty interesting group, the carnivorous plants. So we can look at this. The carnivorous plants probably evolved over the last 90 million years or something like that. We don't really have to pay attention to this very specific set of relationships here, but what we decided to do is we decided to dig into this a little bit more. We decided, instead of doing one big analysis on this thing and generating a tree, taking each of the gene regions, constructing trees of those, so doing this thing that actually requires very little time and very little computational resources, and just look at what data we actually have underlying this. And so we can summarize this in a pie chart. So here we have a pie chart where gray means there's no information in a particular gene to suggest anything. Blue means it agrees with the tree that I'm showing you, and just any other color means it disagrees. Right? So we can see now that actually most of those edges of the tree, there's a lot of agreement. All the genes kind of say the same thing, and there's a lot of information, but there's one that pops out and says, no, this actually disagrees. So we don't know what's going on there, but we can actually now say we understand what's going on. We understand that there is an issue, and we understand where it is. Now, why could this be causing this problem? Well, it could be a lot of reasons. It could be hybridization. That's actually what we think is going on. Ancient hybridization, maybe 50 million years ago, there was some weird hybridization that went on. There could also be horizontal gene transfer, genes moving from one species to another. Could just be noise. Turns out, though, this is everywhere. So if we look at bees and ants and how they're related, same pie charts. Turns out if you look at a bunch of genomes of those, that there are some relationships we know really well and some we just don't know. Turns out that actually if you look at an individual gene, so you go into one gene, that there can be multiple relationships there. So here, this is a gene within primates, and we have for half of the gene, where the red kind of goes above on the y-axis, we have the humans are more closely related to chimps. And then it switches, where the blue is above, to suggest that humans are more closely related to gorillas. So that's within a gene within the human genome. So one question is, can we actually take this kind of analysis and figure out which genes are actually supporting those blue area? Which ones actually have the kind of data that we need to figure out this aspect of our particular question? Because if we could do that, then we could actually try and answer what are those deep nodes in the tree. So, where it's possible, we're trying to simplify this question just to say, I'm interested in whether A is more closely related to B than anything else, and I don't care, and these two trees that I'm showing, E and D have different relationships. I don't care about that. I just want to know whether A is more closely related to B in this gene and whether there's information for it. And we use probabilistic methods to do this. So we have likelihood equations that describe the molecular evolution of this. And so now we've taken our big monolithic analysis, and we've done divide and conquer with this as well. So we take each of the individual genes and we ask that question. Basically, we ask an isolated question of whether something is more closely related to something else. And so what I'm diagramming here is basically the density of the likelihood equation. So when it's really dense and peaky, that means we have a lot of information. When it's really flat, it means we don't have very much information at all. So now, because we can start to resolve these old relationships more confidently, instead of one enormous analysis with hundreds of genes and hundreds of taxa that takes months to run, it's unclear where the signal comes from. There's heterogeneity in the data that's not accounted for well. Now we're conducting hundreds or thousands of analyses on individual genes, asking specific questions. These runs take seconds to minutes. We can take advantage of heterogeneous resources, and we can pinpoint what genes have what information. So this scales easily, and it's reflected in the biology. So heterogeneity is in the data and allowed to be highlighted by this type of analysis. So basically, our field is transitioning, and that's the field of basically genomics, is transitioning from the necessity to have these huge boxes that have, half a terabyte of RAM, multi-CPU, they cost, many tens of thousands of dollars, to something where we can take advantage of distributed resources. And so the reason why we can do this is biological, so not forced by the computational limits, but actually it reflects the biology. So there are a couple of packages that we develop in the lab. One is called PhyBorg, and one is called GoPhy. It's a mix of Go, C++, and C code. Some of it works with WorkQ out of Notre Dame. And this allows us to use our mixed resources to the fullest. So we can still use those 40 terabyte boxes, for those terabyte RAM boxes for the analyses that require them, but we don't require it for everything. So it allows us to conduct analyses really quickly. So let me just give you a couple more simple examples, and then I'll wrap up. So one question that we have in biology, I told you how to start a fist fight for animal biologists. This is the way to start a fist fight for plant biologists. If you went back 100 years and you said one of these alternatives, you would definitely get in a fight. The question is basically, was the first flowering plant a tree with small flowers, or was it a water lily with large flowers? Right? So if you're thinking, what is the timing of that? About 150 million years ago, there was the first lineage of flowering plants that led to eventually about 350,000 species that are essential to all of our life. And we don't know whether it was something that had a large flower and was kind of more similar to a water lily, or something that was like a tree. So we have these two options. So the plants, if you're wondering, one is Ambarella, that's the tree, and one is Nuphar or Nymphaeales, which is the water lily. So we can apply this analysis. We can actually go through, we can take a data set. There was a data set generated that was sampling 1,000 different plants and looking at genomes of those, and try and ask whether or not those genes have enough information to address this particular question, this very specific question. So for example, here are two genes that support the tree being sister to the rest of the plant, so the tree being ancestral. Here is one gene that supports actually the water lily being ancestral. So we can do this across all of the genes, and we can answer definitively that the tree is ancestral. So the tree with the small flowers. So that's that. So these analyses that we did on this, instead of doing one monolithic analysis that takes months, this actually can be distributed and run on a bunch of laptops in the lab or things like that. And it's not just plants, despite the fact that I focus a lot on that. For example, if you do one large analysis on a recent set of genomes that came out of the animal labs, you get that turtles are more closely related to crocodiles than to birds, right? So that's a particular statement. That happens to be wrong. So if you do that one monolithic analysis, you'll get the wrong answer. The reason why you get the wrong answer is because underlying all of that data is a lot of heterogeneity. And so the correct answer is that crocodiles are more closely related to birds. And you get that if you do this other type of analysis, where you look at the individual genes and do a divide and conquer. So essentially, this is how we address this specific question. So we said, in order to do analyses that had thousands of species, we needed to stop doing them all in one big analysis and do these sub-analyses with individual analyses of thousands of species, where we assume the backbone from something. And then how we assume the backbone is we do these analyses that I just showed you, where we look at hundreds or thousands of genes, and we do a divide and conquer on that as well. And that allows us to pinpoint what, where the data lie for each of these questions. All right, so the heterogeneity that we see is real, and it has biological implications. So I just want to leave you with an example of this. So this is a very large phylogeny, many thousands of species that represent that Caryophyllales that includes the cacti and the carnivores and all that kind of stuff. So this has all of that. The colors correspond to the temperature in which they're found, the annual mean temperature. So red means hot, blue means cold. So first off, you can see a lot of heterogeneity there. So there's a lot of evolution from warm to cold environments, basically. There also, I am not showing it, but a lot of evolution from wet to dry environments. And it just so happens that one reason why we're getting all of this heterogeneity and one reason why we're getting all this uncertainty is because that a long time ago, in a lot of these lineages, where you see the black box there was a gene duplication or a genome duplication. So the entire genome duplicated. So we have 2 copies of our genomes. In plants, they can often duplicate their genomes. So what's happening is that every single time there's a really big genome duplication, there's actually some adaptation that seems to be associated with it. So when carnivory evolves, there's a genome duplication. And then there's a lot of conflict that we get and a lot of heterogeneity in those results. So what's really interesting is that we actually are finding that all of that conflict that we're finding that is forcing us to move from monolithic analyses to these divide and conquer analyses has a lot of biological meaning. So I should also say that this analysis is around a gigabytes worth of data, and the runtime takes less than a day, as opposed to when we were first starting out and it taking months. All right, so just to review here, larger data sets yield insights that are impossible from smaller data sets. So this is just a general data science kind of conclusion. It's not particularly novel, but we're certainly finding that in genomic data these days. However, with more data comes more heterogeneity and more complexity. So we can't really get away with doing really big analyses just blindly anymore. We have to do something else. Now, luckily, there are a lot of computational methods that we can take advantage of that can push us from doing these large monolithic things into doing smaller analyses that allow us to take advantage of heterogeneous HPC resources as opposed to trying to find enormous boxes with lots of RAM, et cetera. So this allows for flexibility of computation and all this kind of stuff, and dramatically reduces the cost of entry for genomic computations. All right, so this is the only way in genomics that we're going to be able to continue to do analyses. We're going to soon have petabytes worth of data. We're already generating data that's of that scale. And we're simply not going to be able to analyze it without doing the kinds of methods that I'm demonstrating here that both reflect the computational and biological complexity. All right, so with that, I would just like to thank all of the folks that I work with in my lab and then the several funding sources, National Science Foundation, University of Michigan, and all of my coworkers, colleagues. So, thank you.
Great. Thanks, Stephen. We have a few minutes for questions. There are 2 mics. Actually, there are 4 mics, 5 mics I've seen. Any questions for Stephen?
Yeah, I have a question. How do you deal with the animals and plants that do not have any DNA data available? Everything that's gone extinct. How do you do those analyses?
Yeah, that's a great question. So what we do is we look at morphology. So for example, there's somebody in my lab that uses machine learning techniques to try and get measurements off of either CT scan data, or if you're looking at plants, you have flat images from museums. And so we try to automate measurements from those. And then we do analyses using continuous data instead of the discrete data that I was mostly focusing on. It has different implications, but it's extremely important because it's basically the only data we have for those old relationships. So that's how we do that.
Another question would be, do you think quantum computing could help in this area?
Yeah, I don't know. I don't have access to those resources, but if somebody wants to give me access to those resources, I will answer that question.
I've heard that a lot.
Any other questions? And I'd like to say thank you. Before the next one, I learned to say Carnivorous. It's a good word to brag with my kids.
Hello. Yeah. Do you have a simple relation between the number of the required genes to do the analysis and the deepness of the tree?
Yeah. So there's not a simple equation because it depends on the shape of the tree. So for example, if you have a balanced tree versus a pectinate tree, it's going to be different. The requirements are going to be different for resolving that. However, we can calculate based on a given tree what would be required and basically what the optimal state space would be. So for example, I talked about DNA, but if we looked at amino acids, you would have a different state space, which allows you to have more information in just information theoretic realm. So it's not an exact equation because it depends on the shape of the tree, but you can do it based on simulation, basically.
Okay, thank you.
Mm-hmm.
Looks like that was the last question. We have a gift for you from our conference chair, Michaela Toffer. Thank you so much again.
Thank you very much.
For your talk. Thank you.